Okay, you all. So as we get started, there is a group of core vocabulary here on the left-hand column that you do need in your notes. So this is an applied bit of information, but the good thing about all these terms is that these are phenotypes that occur sometimes because of accidents or injuries, sometimes because of a genetic disorder, sometimes because of other kind of mix of conditions. So these terms here, from ataxia down to strabismus, are things that you'll see as an accumulation of phenotypes that result from having certain kinds of genetic conditions in the examples that we're going to talk about. But one of them alone is not a sign of any one particular outcome. So please hear ataxia. Ataxia is the first one. Crypt orchid. Third one, dysplasia or dysplastic. The fourth one, gynecomastia. 5, hypertelorism. Hyper, as I'm sure you're already familiar with, is going to mean when we're looking at something that is too much, too strong, or above. It means more than in some value. Hypotonia. Hypo, not enough, not a strong enough. Less than or below. Like a hypodermic needle goes below the surface of your skin. So hypotonia. Microcephaly. Microcephaly. Micro means not enough. So very, very small, not the standard. Simian crease. And then the last term here for us in this lesson, strabismus. So do these right now so that we don't have to linger on the slides when we get to the examples for that. So in your reading, be sure that you take a look for and slow down big time when you see any place that's talking about mutation, and then we'll have it also in this presentation today. Mutations are first produced or introduced for you at the end of 3 when they start talking about things that happen during cell division. So you've got some of that on page 50 and also on page 48. Then you have a big breakout section on mutations on page 68 and along in chapter 4, and then it's a huge part of the conversation in 5. Can't overstate the importance of thinking about mutations because we're really heading towards being able to understand, describe, and have the terms for how populations become genetically diverse. So as I put it in the chat, because natural selection only works when you have variable characteristics in your species population. Sources of mutation, again, quite a number of them are really called out in your reading. I jotted a couple of ideas and we'll have a couple here in this presentation. And then levels of mutation. So today we're also going to take a look at the difference and definition of a point mutation with an example of a well-known genetic disorder or condition that's caused by a point mutation. Abnormal chromosome structure. We're going to look at 3 examples of abnormal chromosome structure and see what the outcome of that tends to be. And then the third one, abnormal chromosome number. When something much more major goes on and an individual has more chromosomes than is normal for their species or not enough chromosomes for their species. Usually abnormal chromosome number in humans is not survivable and would result in miscarriage, but sometimes it depends on the chromosome. Sometimes the individual has normal longevity, a human appreciation and fulfillment of their lifespan and so forth. It really just depends on too many, too little, and what chromosome, and whether it's an autosome or whether it's a sex chromosome. Okay, so here we go. Mutations. whenever any cell divides, mutations may occur. And that's just because as the chromosome copies itself and then lines up with its copy in the X form, which are the chromatids attached at the centromere, if all that doesn't go smoothly, then even for somatic cells, there can be changes. However, this is the big one for our class. If we're looking at cell division that produces a gamete, then mutations always occur. And this isn't an exaggeration or me being emphatic. We know that all gametes carry mutated chromosomes. So meiosis is one of two types of cell division, and that's the type that produces a gamete. So you've got a couple pages of reading and diagrams in 3. There is an entire page in Canvas devoted to the video on meiosis, and also asks you to jot down the statistics of how many different combinations of chromosomes you can produce within one person or with another individual. So they always occur. So meiosis always produces a mutated chromosome. So you and I and every other individual organism on the planet that's the product of sexual reproduction, and that just means there's a gamete and it has to meet up with another gamete, and that includes the majority of plants, for instance, fishes, humans, insects, etc. We are all slightly mutated versions of our parents. So diversity and variety is always being introduced to our populations. But for us in class, reproduction, who's reproducing with whom, how frequent is it, what is being selected for and perhaps not selected for, that is the big concept for the balance of the course. And that's because mutations that occur in gametes play a role in evolution. Mutations that happen in your somatic cells do not play a role in evolution. And that's because your code was set when you were created as a fertilized ovum, and then that code stays the same for the rest of your life. If you get a mutation in a somatic cell, say for instance, because you've had too much of a certain type of radiation, you end up with a condition we call cancer. Those cells stay in you. You can't pass them to the future. Since they don't go to the future, they don't play a role in evolution. So really, it's just what's going on with the gametes that we send forward. And that's why also, when we were studying this, we were only looking at the concept of fitness being related to who actually does reproduce, not who could or who had an offspring but that didn't survive for very long, but who actually passes their genetic combination to the future to a grandchild generation. So gametes pass to the future. So anything that's changed in there goes into the new population gene pool. So they persist. You might have heard people say some version of this in many different languages, that children are a form of immortality. And that is correct. It is the organic part of you that can keep going. The thing about mutations, though, is in the DNA molecule along the length of the chromosome, there's no place where it's more or less likely to occur. So it's really random and unpredictable, at least as far as data that we have right now, to figure out where it might occur and what the outcome might be. The ones that matter the most, though, to evolution are certainly going to be those that are in the exons with structural or regulatory functions, because those are going to make the difference between whether the offspring is going to be able to function just fine and become a parent and reproduce, or perhaps not live long, or live fine but not be a parent. And occasionally when you're reading about this a little bit more, you'll see the phrase germline. Germline means we are tracking the cells that go to the future. So that's what germline is. All right, the result of mutations. One of the interesting things about thinking about mutation for the understanding of it in the entire general population is that the general public knows what the word is and knows that sometimes things can really go wrong. In fact, actually, the majority of people, when you say mutation, think that that means that all the time things really go wrong when you have a mutation. If you look at this just socially, for those of you who have other majors like in media or in literature or in things that might be more popularized for the public, probably the public's awareness of this comes from mid-century in the 1900s and is mainly associated with the invention of atomic weapons. Not very much information was passed to the public, but all of it was incredibly scary. They knew that they were incredibly destructive, but they also knew that as the after effect of the use of them against the Japanese public is that not only did it destroy buildings, but even for some of the people that survived, within a short period of time, they had massive genetic changes that were causing them to pass away in horrible conditions, particularly cancers. And then of the next generation, for those that didn't pass, it was also still building up, that this mutated genetic combinations were causing cancers and other conditions to be more pronounced in the population. So it was scary, it was ugly, it was evil, it was mean. It became a very, very frightening thing. Once the technology was out, then it became something that even for kids during that particular mid-century period of time, that instead of having that like horribly scary drills that we have now in public and some private institutions for little kids, the active shooter drills, where the kids are just frightened to death and they're getting ready for something out of their control to happen. The same thing was happening, but with a different type of drill. And they were having bomb drills, atomic weapon drills, big mushroom cloud things. And even though nothing that the kids did or the school would have done would have saved their lives, because this was this unstoppable force, they would be hiding underneath the desk, covering up and just be terrified. That terror flowed over into creativity. And we see that it's about that time where we start to see more of these post-apocalyptic visions in literature or movies, the sci-fi movies, horror movies. Zombies are taken as a concept from a real indigenous activity and concept to something that's related to weaponry and warfare and other kinds of awful, nasty things that humans do to each other. So everybody got some awareness of it, but only when the mutation was deleterious here. So deleterious is your college student vocabulary word for having a bad outcome. In terms of our course, a deleterious mutation is really going to be something that makes it impossible or harder or not as successful for that individual organism of their species to produce offspring. So think of it this way, for our course, when we're looking at evolution, a mutation that makes it so that maybe a portion of your physicality isn't formed exactly the way that it's standard, or maybe doesn't work quite as easily, if it still allows you to survive, be reasonably healthy, and live long enough to reproduce and parent effectively, it isn't actually, in terms of natural selection, that deleterious. So a human, for instance, that's born with extra fingers or missing fingers, or a human that can't hear, for instance, because we're a social species and our community fills in the blanks for that, that really wouldn't be a minus to your ability to reproduce. So a deleterious mutation, perhaps you're not making any sperms, perhaps you can't digest your food, perhaps there's something else going on so that no one would like to be your reproductive partner, or they don't choose to do that. So deleterious mutation. So that means bad.
So
for continuing your line. Next one, neutral mutations. A neutral mutation is actually probably the most common outcome of all genetic mutations for any species. And that means that whatever the code that was changed isn't making any difference in the proteins that you need or that you're producing that help you to stay alive and live and function. So if it doesn't have any effect on reproduction at all, and the individual lives long enough through their reproductive max time, then it's neutral. We do know that this is probably likely statistically to be the most common outcome of most types of mutations. We just notice it when it's the other ones. It also is true that mutations can be beneficial. So there are kind of a lot of examples of that if you wanted to look into it into one more detail, but I'll give you a quick example before we move on. Beginning in about the 1980s and getting closer to 1990, there was a lot of global research on the human immunodeficiency virus. It wasn't new on the planet, it was already present in other species, but when it finally got into humans, it caused major disruption in human lives. It didn't really actually cause major disruption in other particular organisms, which is something we'll look at later on in the course. So while they were looking for this, they were trying to figure out, most specifically, method of transmission and how dangerous it might be to have a coworker or a parent or a neighbor or something else, because it was a virus and they had to figure out how viruses were passed. When they started to look specifically within households of people who were actively exhibiting HIV to their sexual partners who were with them and were having intimate contact, they found in a few small places, one of the populations was in a very small region in Italy, where there were quite a number of individuals in this small town region that had been exposed over and over and over again because their long-term partner or spouse was positive and they never caught the disease. And they found out that every one of them had a oddball mutation that made it so that the human immunodeficiency virus couldn't get into the right cells. And so they were immune. That's a beneficial mutation at the time that that disease emerges. So mutations can be beneficial. Thinking about neutral mutations though, because this helps us to review something that we looked at today and also on Monday, because about 75% of your base pair sequence is non-coding. If there is a mutation within that region and it doesn't turn anything on, then the outcome is neutral, right? Nothing has changed. It hasn't made any active code that's making a structural change or a regulatory change. So if the mutation is not changing your phenotype, practicing some more vocabulary here, then it is neutral. So mutations in non-coding regions, or if they don't change the amino acid sequence. If you remember from last time, when you look in chapter 3 at the amino acid table, some of those amino acids can be spelled 6 different ways. So if the base pair sequence makes a small change, but it still makes the same amino, then you're getting the same protein and everything stays the same. So that's what a neutral mutation would be like.
I have a question.
Yes, go ahead. Go ahead.
So like a neutral mutation is just talking about whether it affects your phenotype and how it goes, right?
Right.
So would that mean that, let's say, coronavirus itself, since it's a mutation, does that mean it's a neutral mutation?
So coronavirus itself, yeah, is something that has a mutation from something that was already present. And that mutation for coronavirus has made it very easy for people to catch. So for actually whatever that mutation was for that species, right? Because viruses are their own living things, they're their own genus and species. So that was kind of a plus for the coronavirus to make the change that it is. Because when it makes us cough and sneeze, and because we carry it for a while before anybody will know that you're sick. So that was good for that species. It hasn't been good for us. So we so far haven't seen where that's going to end up for that. But yeah, every organism gets its own mutations. Is that kind of what you were asking towards?
Actually, I was just wondering if like what coronavirus be considered a neutral mutation, or like is there a good or is it a bad one? I'm just
Oh,
curious.
for humans, it is an agent of natural selection right now. And it is thinning out some segments of our population. So it is a natural thing. It's not happy, but it is a natural thing that happens in nature, particularly when any species becomes extremely overpopulated and really crowded, right? Which is why we saw it tear through big urban environments first, and where we see that some places in the world, or even states in the United States, where there aren't very many big cities and people live really, really separated, where they're really not having very much transition for that. So coronavirus as a mutation is probably not quite the thing to think about with it, but it is an agent of natural selection. And at first they were telling us all, remember back in March, that young people didn't need to worry about this at all. It was just really, we were trying to close down and separate to protect our loved seniors and older people, or people that might have some other health issues in our population. But it turns out it is impacting certain members of our younger population. And it looks like it might have some neurological or actually even vein blood transfer kind of thing. So yeah, it has a natural place in nature. Once it's done making it cycle in the future, there probably will be less of us that have the strongest effect of it, other than people that already have some health issues. But one of the health issues that they aren't really saying about it is obesity. So people whose respiratory and heart system is already under a lot of pressure because they have a lot of body mass are having difficulty if the not the best way, but nature does bring populations down in every species when they become overpopulated. And so since we don't manage it and space ourselves out and manage our birth rate around the world, then nature will do it. It's the same thing that happens with rats and weeds and other kinds of stuff. So there's happier ways we could manage this. Okay, so fitness, again, really practicing for this unit and all the rest of them. Fitness is whether you're going to pass along your characteristics to the future, to a new generation. So it can affect anatomy or physiology, but not have any strong role as to whether you're going to reproduce. So for instance, if you have a mutation that doesn't make you really falling apart or really sick or really not able to function until you're 30, well, you still had between about 15 to 30 to become a biological parent. So that really isn't inhibiting your opportunity to pass your genetic combination to the future. So that's kind of the idea of neutral here. All right, so we're going to look at, now we're switching over. If you go to a new page or go down a few lines, we're going to look at point mutation and then chromosome structural mutations before we are wrapping it up for today. So we're on this one, just this one that's brighter in white, point mutation. We're going to look as an example of a point mutation at sickle cell disease or sickle cell condition, sometimes called sickle cell anemia. This depends on kind of who you're talking to and whether it's really medical or how the conversation is. There are lots of other examples of point mutations, but this one is been reasonably common in the global population for as long as we've been looking at these kinds of disorders. So we're going to define what a mutation is and then take a look at how these can pass along. So point mutation. Okay. Now, contrary to what a lot of people in even urban American society think, sickle cell disease is not something you catch from another person. It is something that can be inherited, so it can be passed from a parent to children, but here's the third part. anyone with this mutated gene has the disease. So you can either get it as a mutation, and it will be present then from when the baby's born, or it might be inherited. It is not only a portion of the African population or the Black American population. It is a fairly common genetic condition in Africa, South and Central America, Caribbean islands, India, Saudi Arabia, and right in that warmer part of the Mediterranean, Turkey, Greece, and Italy. Basically, this particular mutation affects just one thing in your body, and that is the ability of your red blood cells to withstand pressures that happen when you have low oxygen density. So this little girl here, Tiana Hughes, was one of the US child ambassadors, isn't the current one, but was before. And as you can see, she doesn't have any really horrific things going on with her at the moment. We have very good treatments for this nowadays. We've got, if you've got insurance or you have access to blood transfusion and some other types of treatments, they can really make a big difference. And not everyone who has a copy of the gene even actually knows that they're carrying a copy of the gene, because their symptoms can be fairly mild. So under the microscope, what happens with this mutation is it causes red blood cells, which here should look like this all the time, no matter what, even if you're exercising, even if you're sick, even if you're exhausted, to look sort of like a little breath mint that has a little dent in the middle or a pillow. When you have one copy of the sickle cell gene, then what happens is under certain stressful physical situations, this warps and takes on this sickle or crescent shape. When it does this, it can't do its job as well or at all. So we're going to investigate this a little bit more detail here. Okay, so basic definition of point mutations. And it helps us to practice some terms we've been working on for about a week. So a point mutation occurs when one base in the cell changes. So only one nucleotide has a change. You got 3 billion bases in your system. So there's 3 billion spots where point mutations can occur. But we don't know about it if there's a change unless it's in a place that's got active code, and then that code is altered. So sickle cell disease is caused by a point mutation, which is one nucleotide changes. And in that particular case, the code for your polypeptide chain, remember polypeptides, that's the chain of amino acids. The triplets turn into codons and they make the amino acids, is altered. And it's got to be in the sequence that makes hemoglobin, which is another form of protein, and we call that red blood cells. If an individual has only one copy of the gene, they don't have two copies, right? They don't have both alleles the same, but they just have one of the alleles, then they will have much milder symptoms, to the level that it's possible in many cases that they never actually find out that they even carry this gene. However, if you have two copies of it, you will know it. The child will know it right away. It'll be very, very obvious, and they will need assistance, treatment, and mitigation. So as an example of when you have just one copy of the gene, right? When you're heterozygous for this mutated gene, my example person here is, even if you have never heard of this man, you know from his outfit and his general physicality that he's insanely fit. He was a professional football player in the NFL for quite some time, the Giants, Steelers, and Redskins. This is Ryan Clark. He was a starter and We all know how often you get injured, how exhausting it is, how much exercise and time you have to spend in physical training. We know that to be able to play like this in the NFL, you started when you were much younger, yet he actually never had any type of noticeable event until he played a game at the high altitude at the Mile High Stadium in Denver. At that particular point in his life, as a big, super healthy, super energetic human, he actually had an attack and had to go to the hospital. What happened was that the oxygen deprivation that was happening, because at altitude there isn't as much oxygen as the air, was stressing out his blood, and it was stressing out everyone else's too. But because he had one copy of the gene, a portion of his red blood cells sickled, and that caused something that's known as an infarction. Infarction is a really scary term if you hear this at the hospital or you know someone that's had this as a diagnosis, because infarction means that your blood supply has been obstructed. So when those cells warped, then they caught on each other and they blocked oxygenated blood from coming in and out of his spleen. So he did recover. It didn't end his career. I think he's currently now coaching and doing some other things. And as the awesome person that he is a founder of Ryan Clark's Cure League. Last time I looked into this, they help with funding, not for research or not from the sciencey part, but actually funding for families who need financial assistance because they don't have insurance, or they might not have transportation, or they just need help finding doctors and finding treatments and then paying for it. So yeah. So there are other celebrity individuals that I could have picked, but it's really easy. We all know how often and how physical that particular career is. So one copy of it is not something that's going to end every part of your life. 2 copies, much, much more dangerous. Now, hopefully this is going to run for us just fine. You should be able to hear this. This is only a minute. It's going to talk about what happens and why it's called sickle cell.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need.
So here's your DNA
Sickle
strand
cell anemia
unzipping.
is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
So very frequently, you'll see photos of people who have 2 copies of the gene, of the affected gene, with really inflamed joints. And that's because the blood vessels are very narrow and small there, and then everything backs up. And then when they aren't getting oxygen, then tissues are dying. So you've got some really awful things going on for people that have 2 copies of it. Okay, so to practice again, I'm not going to ask you specifically what chromosome it is, but just in case you know someone or someone in your family or you've come in contact with it, this is 1 base on chromosome 11. And on that one, right, a point mutation occurred. The T that's supposed to be there was substituted out for an A, and in that case, it is not making the right amino acid there. And when that is like that, then when it builds and produces the red blood cell, then they can't flow constantly the way they're supposed to. So again, a couple more vocabulary terms. So here, if you have 2 copies of the standard gene that's going to make the T here, then all your blood cells stay normal and round and don't clog and do carry oxygen. If you are heterozygous and you have one of each, then under certain kinds of intense stress, you might have an attack, but still most of your blood is carrying oxygen the way that it's supposed to. But if a person is homozygous for the non-standard allele, the A allele here, then under any kind of stress, all of the blood cells sickle. They're not carrying oxygen the way that they're supposed to. It's extremely pain, swelling because tissues are dying, and it's really, really dangerous condition. So you can inherit it, or you can be born with the mutation that just happened and not have it come from either parent. If the parents are like this, though, both of them could carry the gene and perhaps not know that they're really affected and then pass it along to the offspring, perhaps. Okay, so here it is one last time. So here it is, point mutation contains just one base in your base pair sequence, but that base is part of the triplet code that makes one of the proteins that makes red blood cells. All right, so you don't need glutamic acid or valine here, just the basics. If you have one copy or two copies, it's a point mutation, and that means it changed one base in the code. All right, so a couple more terms here. Practice a little bit, and then also, because you might read about this if you are going into something that's healthcare related, like even the business of running healthcare or billing of healthcare, or nursing or doctoring, or laboratory work, analyzing genetic samples, blood samples, all that good stuff. Autosomal recessive diseases. There are a lot of them that have been recorded. So first word here from Monday, autosome. Autosome just tells you where. It's somewhere in the 1 through 22 chromosome pairs, right? Your autosomes are number 1 through 22. Those are the ones responsible for the structure and function of you or any other organism to be able to stay alive. If the next term says recessive, then it means that the allele is not all that powerful, and so you will not show the disease or the outcome unless you have 2 copies. So all recessive situations require that you have 2 copies. And 2 copies means you are homozygous for the recessive. So here's something to put in your notes, a little star or a little highlight. Recessive doesn't mean less healthy. It doesn't mean less desirable. It doesn't mean that it isn't completely standard for the species. Recessive just means that it's not making an exceptionally powerful protein, and that there might be a variant that is more able to express itself. If there is a variant that's able to mask the weaker one, then we say the variant is dominant. So recessive doesn't equal healthy or unhealthy, and neither does the term dominant. Dominant doesn't mean that it's a healthy variant or an unhealthy variant. It just means that if you have 1 copy of it, you know it. You know you carry it. It's going to show up. Now, with recessive disorders, they tend to, the genes that cause them, they tend to hide in the population and pop up and down over time. There's another one, it doesn't cause the same effects as sickle cell, but it is inherited in a similar way, and it's the same kind of situation. If you have 1 copy of the sickle, of the Tay-Sachs, so Tay-Sachs disease, if you have 1 copy of the Tay-Sachs gene, then you don't know it, nothing's really changed. But if both parents carry 1 copy of that recessive Tay-Sachs and the baby is born with it, then the baby is immediately from birth very, very ill and has a very, very short lifespan. So it's really sad and really, really difficult for the parents and the family because they are going to lose their baby fairly quickly. If they have one copy, then the parent is a carrier. And that's what the word carrier is. You have a recessive that might be a problem, but if you only have one copy, it isn't interfering with your health particularly, so you might pass it on. Even so, if both parents are carriers, right? So they have one of the standard and one that's not so desirable, one of standard, one that's not desirable. In each pregnancy, it's only a 25% chance that the baby born in that pregnancy will actually be homozygous for the problem alleles. So they only have a 1 in 4 chance of having a baby that's affected. Now, last one down here, dominant disorder. A dominant disorder is always noticeable, always able to see the outcome or detect that something is different, because it means that if you have one copy of a dominant gene, it's going to show up. You're going to know. Like when we looked at blood before we went to break. If you have an A or a B, then it's going to show up in your blood type. So dominant disorders are always very clearly present in the family line, that somebody's got it and they might be able to pass it along. Recessive genes hide, and sometimes it's just not clear whether anyone is affected or carrying that. Okay, now go down a couple lines. Let's look at chromosome structural abnormalities, and if we have time, we can take a look at numeral abnormalities. So we'll do structural changes first, and then numeral second. Chromosomal structural abnormalities mean that a whole bunch of the base pair sequence has been changed. It's not just one base or one nucleotide, it's multiples. And there are 4 ways that this generally can occur. The outcome of a structural abnormality depends on which chromosome is affected and what that chromosome does, and also how much of its base pair sequence was changed. In numeral abnormalities, it means that the hugest effect has occurred, that the individual has gotten an extra chromosome or is missing a chromosome. The outcome of numeral abnormality is profound, but the outcome depends on which chromosome is extra or which chromosome is missing. When we looked at karyotypes on Monday, one of the karyotypes that I showed you right off the bat had a red circle around some chromosomes, and in that circle was enclosing 3 chromosomes at that position. So that's a numeral abnormality. So people with these do have a strong effect if it's structural or if it's numeral. Okay, so let's look at structural changes. So there's 4 ways that the structure of a chromosome can be altered in larger sequence. And lucky for all people looking at it who don't do this all the time for a living or for your major, what the term says is exactly what happens. So you just have to envision the whole chromosome, right, with its sugar phosphates on the side and the bases coming in towards the center. And then if it's a chromosome structural abnormality because of deletion, it means that the chromosome broke horizontally between the sugar phosphate and a chunk of it came out. It just is missing. Let me see if I can do it isn't that easy, but I think I can do this kind of simply here. So here's your chromosome one side, and that's the sugar phosphate side, and here's the other sugar phosphate side. When we're looking at these, right, you have the nucleotides with their bases coming in to the center and so forth. So as they're doing this, right, the DNA base that's here has its specific partner. If this is a T, then this one's an A. If this is a G, a C. This is a C, this is a G. And we were talking about last time that the sugar and phosphate bonds, oh boy, it's hard to do this with a, won't let me use my pen here. But the sugar phosphates here, these are very strong bonds. These down the center, those hydrogen bonds between the DNA bases are weaker and they're supposed to open up. But if this breaks this way and this way, right, you get a gap in between this sugar phosphate and the sugar phosphate down here. The problem is that sugar phosphate will bond to any other sugar phosphate. It's not picky. So if this breaks here and here, and it floats away, then the part that's left up here is just going to re-click down here, and you have deletion. Sometimes what happens is it breaks, or something, there's a gap, and it goes through a repairing kind of process, and it just adds more sugar phosphate, sugar phosphate, sugar phosphate. That's duplication. Duplication is when it adds more code here than was there originally before the chromosome broke. But again, the breaking is kind of from the diagrams we've been using in the horizontal between the sugar phosphate. Inversion is an interesting one to think about, and they have actually been able to find that. With inversion, it breaks here and here, but remember, the cytoplasm is liquid and it's three-dimensional. It's too tiny for us to take a photo of up close right now, but it is three-dimensional space, sort of like something floating in, let's say, an organized fish tank. So this comes out and it flips upside down, so that if this started off as an A up here, now the A is down here. So that's inversion. It glues it back in, but it's not in the right sequence. So that's inversion. And translocation, sometimes this breaks and then just swims off to someplace else and glues someplace completely unrelated. There are examples of it breaking and gluing on to another chromosome where it doesn't belong. What's the end result though? The generic end result for us to think about right now at this particular part in our lesson? The end result is, let me see if I can get some of these to go away. Go away. Go away. Okay, and let's make this one go. And this one. The end result for this is that when RNA comes into the nucleus to read the DNA molecule unzips a little bit, like right down the center here, and then RNA comes up and reads a portion of the sequence. If some code is gone, RNA goes to the address, but now what it's looking for at that address is changed. It copies, edits, and carries it out. It's making the wrong thing. If it's duplicated, same problem. All the addresses are wrong in the direction that it's trying to read. If it's inverted, the spelling of the word that it's trying to make is upside down, doesn't work. And if it's missing and it's not even on here at all, then it never even sees it. So all of this disrupt the code that RNA reads and makes it so that it cannot assemble the proteins that it needs to assemble. All right, so we are just going to look at deletion mutations. So we're going to look at and use some of those terms you jotted down in the beginning, ataxia down to simian crease and so forth. in terms of deletion mutations. And I'm going to give you 3 examples. We're going to look at 3 examples, and you'll be able to really see how, depending on what's lost in what way and on what chromosome, how you get really different outcome for the people that are carrying this genetic combination. So deletion, again, when more than a single base pair is deleted. Of course, the more that's missing, the bigger the outcome will be in the phenotype. So first example, please do write down deletion disorder, and the name of this one, cri du chat. It means cat's cry in French. I probably didn't pronounce that even close to correctly because I never took French class. This is very rare. It occurs in maybe a little bit more frequently than about 1 in 50,000 live births. When the babies are born alive and there are kids, what is the locus? So in this one, and here's a karyotype over here, again, practicing the concept. Here's a karyotype. So a baby's born and has a couple of unusual things going on. And so there might be some idea that perhaps this is genetic, not a bacterial infection or something going on with the mother's milk or something else like that. So they go ahead and do the karyotype. And what they'll see is over here, right? Autosomes have numbers 1 through 22. So they're looking in the autosomes. And then here you can see on this one, here's the centromere. You all, I'm practicing as many vocabulary words as I can with you. Centromere right here, the little pinched waist. So on the P arm, short arm, the P arm of the centromere, you can see on one of these chromosomes, this dark banded area. So there's a light one and then dark and then the rounded tip. On this one, it's gone. It has been deleted. So if we look at this even closer, you can see that there's a chunk up here that's missing, or in a diagram, it's missing up here. So cri du chat. So missing bit of chromosomes on the short arm of chromosome 5. So the outcome is going to depend on what that sequence of code is supposed to do on chromosome 5. And the second part on how much broke off, how much is missing. So, okay, this is self-posted by the family, and there's a narration really going. You're just going to be able to see this little adorable baby. You can see right away that he does have a change in the code. His coding got confused, and it did not fuse the skin here all the way across the upper lip. So that is a form of dysplasia. We might have some popular names for that lip splitting. That's not by itself particularly super rare, but dysplasia, it just means tissue that didn't form in the standard way. But let's look at some of the other things going on with this little one. So that's the baby's voice.
Yeah.
You can see as he's growing up, he gets some surgery to correct his upper lip. Oh, little one. And he's getting a little bit of corrective surgery on his lens.
Are you a hungry monster?
And then a little bit later on, like regular kids, wanting his earbuds and his toys. That was posted by the family. So I would never take something from somebody that, there we go. Yikes. Okay, so I don't know why that went backwards. All right, so again, this is called cri du chat, and obviously this would be one recognizable from a long time ago, because generally that's not what babies' voices sound like when they are first born. So first use of the vocabulary words then that you jotted down, hypotonia, hypotonia. These little ones are born with low muscle strength or muscle weight or tone. That's a better one, muscle tone. And so they're very weak. So part of the reason his voice sounded so high-pitched, like a kitten cry, instead of a human baby belting it out, is because they're very weak. That can happen when the mom's not well. It could happen from bacteria, virus. It could happen for other kinds of stress or injuries. It isn't by itself only associated with this or even with a genetic disorder, but it is the first clue, right? The first little bit of empirical data. They need to look more closely into this baby's health. Also, these children are frequently somewhat microcephalic. Micro, a little bit smaller. Ceph is your brain box. So the back of the head that houses the brain is smaller than generally what they see in babies at that birth weight. It doesn't also, that can happen from a lot of other different circumstances. And again, it's going to vary. It doesn't mean that the brain is missing. It just means that it's not fully the size they normally see. So they're also generally microcephalic. They do have some very distinctive facial features. And if you just take a look at this little girl's cute face here, we'll see another in a couple minutes or in a couple slides, and take a look at that one. So the dysplasia that we saw on the little boy's upper lip, where the skin didn't get the right code to fuse straight across, also can result in some other confusion about building tissue elsewhere. Sometimes the ears are not set perfectly and they're very low along the side. Or here we can see a little skin tag here, just a little overgrowth of tissue in a place that it's not supposed to be. These several things coming together would be starting to show the helper medical personnel of what to be looking for in terms of a diagnosis. In this little one, this is a different baby than we just saw. The ears are a little bit low set, and you can see here that while the baby does have a lower jaw, it's not quite the length it should be to match the full length of the upper jaw. That later on in life is correctable as well, but it does mean there's an error in code. So can you see easily from just these few pictures that when it's multiple genes that are affected, right? They have one normal copy of the gene, but it can't work with its allele, then you start to have a cluster of phenotypes that are affected, structures or functions that are affected. So for these little ones, you can also see that a lot of the changes are taking place around the face, the side of the head, because that's part of where that code to build those structures on everyone lives or is located. Now, see this little girl as well, beautiful as she is, and none of us would really look twice and think anything if we saw her out in the street or out playing or in our classroom, how widely set her eyes are. So that is called hypertelorism. And it means that all the children, no matter how strongly affected with this deletion disorder, whether they didn't lose too much genes or whether they lost more genes, are going to end up with this little widely spaced eyes, and that is not coming to them from the rest of the family. Some families do. Everybody has very nicely widely spaced eyes. But in this case, her family does not. And we saw the same thing in the little light-haired girl that was in the first slide for this one. So that's called hypertelorism. Does it make any problems with her vision? Nope, no problems with vision, just a slight change to the build. And again, what does microcephaly look like? It doesn't mean there's no brain, it just means that it's not as large as we would expect it to be. That just does generally mean that there are some slowdowns in learning, the learning process, but it doesn't mean they don't learn. It just means that it's going to take a little bit longer. Okay, so skip down a couple more. Let's look at a different example of deletion. Same thing, the loss of a series of triplet code in a particular area. But this time, for this one, I want us to take a look at something that's much more advanced than what Gregor Mendel could have ever studied, and something that until very recently, there weren't that many researchers that actually were particularly aware of it. And that's called genomic imprinting. So in this particular case, we're going to look at two disorders. And again, the people that are affected with them, they have lives. They just have challenges that those of us that are genetically standard don't have to worry about or don't have to work around for this. So in this case, the chromosome is chromosome 15. And again, practicing this terminology, the Q arm. So that's the longer section of the chromosome. So Q arm. So chromosome 15, Q arm. It is deletion, so it means a run of code is missing. It broke off. It's not on the chromosome anymore. If the chromosome that has the missing run of code came from the father, then it produces a set of phenotypes that we call Prader-Willi syndrome. If that chromosome came in the ovum and it was from the mother's side, then the phenotypes that are produced, even though it's the same area that broke off, it doesn't give exactly the same phenotypes or changes. And that's called Angelman. So what does it really mean? It means that incredibly, like I can barely imagine, it means that your body knows. Here's a nice little, my attempt at a nice little sparkle here. Your body knows, if it's all the time, I don't think they can prove it right now, but at least at times, your body knows, and it matters whether the chromosome was in the ovum or whether the chromosome was in the sperm to begin with. Incredibly interesting. So again, what's broken off, what's missing is this gene run further down on the bottom of the chromosome. If it's missing from the fathers, then it activates something else. If it's missing from the mothers, then there's a different one. You don't need this chart, but genomic imprinting, that means that whether the change is on the sperm or the ovum matters, and then a couple of these examples. So on this one, this particular individual, probably all of you recognize, or you'll probably recognize her voice. Mayim Bialik has been famous actor since she was a little child, and then long-running TV show that's been on TV for a long time, and she's very good at all that, but she's also freaking genius, and she has a PhD in biochemistry. Her doctoral dissertation actually was specifically on this deletion area on chromosome 15 that produces Prader-Willi. So let's let her explain what the changes are that happen.
Hi, I'm Mayim Bialik. You may know me as Amy Farrah Fowler from the hit comedy show The Big Bang Theory, or from the TV show Blossom. What some of you may not know is I have a doctoral degree in neuroscience. While working on my degree at the University of California, Los Angeles, in 2007, I wrote my dissertation on a rare genetic disorder known as Prader-Willi syndrome, or PWS. Prader-Willi syndrome is the leading cause of childhood obesity, and it affects approximately 1 in 15,000 births. People with this syndrome constantly feel hungry. They don't have the ability to ever feel full. So without support, this can lead to life-threatening obesity, along with many other medical complications. If not closely monitored, individuals with PWS can potentially die from overeating. In addition, people with PWS suffer from a number of other symptoms, including medical, psychological, and developmental challenges. As an infant, ironically, babies with PWS are very weak in muscle tone and have no appetite, so the parent's effort is spent trying to get them to eat. The child's appetite then typically begins to increase between the ages of 2 to 4 years old, at which time the hunger begins to take over. Imagine every day thinking about food all the time, constantly figuring out when you can eat next or how you can get food. Remember a time when you felt your hungriest? Imagine feeling like that all day, every day, for the rest of your life. Kids with PWS will continue to eat and gain weight on considerably fewer calories than the average person. If not properly supervised, some individuals can gain up to 20 pounds in one weekend. Parents and caregivers often have to lock their cabinets and refrigerators to keep their child with PWS from eating. As one 8-year-old boy said to his parents when he thanked them for locking up food, "I try and I try, but my hand reaches into the refrigerator and I can't stop it." Keeping a regimented food schedule, securing food, and keeping it out of sight are some of the many ways parents and caregivers are able to help minimize the problem and reduce the stress on both themselves and their child. As the child gets older, the food issues in school and everywhere else in society becomes more of a challenge. Sadly, in order to save their lives, parents have to severely restrict their teen and adult child's freedom. As adults, many people with PWS will live in highly structured residential settings, most with 24-hour-a-day, 7-day-a-week supervision. But there is hope. Prader-Willi Syndrome Association USA has been providing life-saving research, crisis and medical support, education, and newly diagnosed family support since 1975. With a national office in Sarasota, Florida, and chapters all around the country, they are saving and transforming the lives of people with Prader-Willi syndrome.
Okay, so I'm going to stop right there because she's going to go on and do some other social commentary there. So again, genomic imprinting, you might sometimes also see that as a sex-linked disorder. That means that the body knows if it came in a sperm or an ovum. It doesn't mean that you got it because you had sex, and it doesn't mean that it comes from the sex chromosomes. It just means that whether it's carried of contact from one of those or another. So yeah, I've seen some other programs on this particular disorder, and it is just, for those that have the strongest version of it, it's really, really
Last year,
because you can barely,
PWSA
barely imagine what it would be like to, see if we can get this to let me leave this, to feel as if you were starving every minute of every day of your life. And that's really what's happening. When they eat, their body doesn't tell, the stomach doesn't tell the rest of the body that it's full. The brain never feels satisfied. It doesn't have satiety. And instead, what it's doing is running that panic thing because it cannot tell that you ate. And so they feel like they're starving. So you did notice that she did quote child talking and interacting. So verbally, fine. Their use of English, full sentences, good coordination generally for most of them, and that sort of thing. This one, again, it's rare, but 1 in 15,000 means it's a lot more common than Cri du Chat. It is not inherited generally. Generally, these are not individuals that someone else in any kind of, okay situation would be exchanging gametes with. So the IQ generally a little bit less than what we normally see in population, but not so low that they can't enjoy other things in life. One of the things that does occur though, so she said they started off with low muscle tone, so hypotonia again. This is a normal kind of typical early on visit back to the doctor with your babies once they're weeks old, where the doctor gently pulls up a little bit and is looking to see that the baby's going to want to pull their head forward, or leans them forward this way and the baby wants to lift the head up and see where they're going. As long as they can do that, then that at the right age, then that means that they're moving along. Just because they can't do that does not mean they have a genetic disorder. Again, the term just means low muscle tone. So maybe they're not feeding well. Maybe the mom's breast milk doesn't have enough, or it doesn't have enough nourishment in it. Maybe it's a digestion, maybe they have an infection, maybe something else is going on there. So, and then the last thing, these two individuals and the woman that you saw during the video sitting next to her dog, they're not related, but you can see how closely similar the shape of the face is. One of the things that's happening with this is it does diminish pigmentation levels in the hair and eyes and skin. So while they aren't albino, they don't have as much variety as we might see in their own general families there. And so for some of them, because they didn't lose as much of the broken section on the Q in the 15. Their hyperphagia, right? Their drive to continue to eat. So that's what phage means, phagia, consumption. So they're driven to eat too much, may fade down to where they're very interested in food, but honestly, that's like our population and our culture at this moment anyway. We did have a neighbor where one of the family members did have this genetic change, and he was very social. He would go on walks around the neighborhood, remembered everyone's name, but not too long into little chat conversations like over the fence or when you saw him while we were walking the dog, did he ask, do you have a snack? Do you have a candy bar in your pocket? Could I come in your house? And we knew his parents were like, oh, your mom's gonna be so mad if you come in the house. And he'd say, I know, she'll be so mad. I can't go in the house. So, you know, did enjoy life to a certain extent for those individuals. But if it's really bad, they do have to live someplace where they don't smell food, they don't see any ads, they're not having any toys that look like a plastic hamburger, where they can just try to forget that as best they can in between the meals. But it does change some of the coding in the face and for pigmentation as well. Now, we're going to skip this one for Angelman. Angelman is the other side of this. So this one is sex-linked or linked to maternal 15 code. In this case, the result is really different. Just about the only thing that's similar between the two, even though it's the same segment, is that loss of pigmentation. When you see photos, a whole lot of them are very fair skin, very, very light in the eyes and hair, even though the parents are not. It doesn't mean they're albinos. It just means it's going to be lighter than what their family passed on. So developmental delays, though, in Angelman are much, much more severe. These children frequently never can speak in sentences. They also have ataxia. So ataxia is a good one for all of us to know. Ataxia is when you have a loss of balance, so that you're not able to neurologically stand up and walk in a straight line and look steady. So the person might look like they're leaning or they're staggering or they can't seem to hold. Now, ataxia is the same word whether you're intoxicated or whether you've had, for instance, an accident that's damaged your brain and you don't have a functional state sense of balance anymore. Ataxia can come from inner ear injuries or disorders, and it can be genetic. So ataxia, loss of being able to hold your balance. So no speech at all or very minimal. They do have neurological problems where they clench up and their nerves don't fire very well, and they'll get their little hands up and they'll kind of be shaky. But they, almost every photo shows them with a big smile on the face, smiling and laughing. Whether emotionally that's the state or not, I don't know that that's super clear from researching it a little bit, but the parents do think that they're happy enough. Highly social interaction. But because they can't walk around and they have really severe some other issues with health, they will never really be going on walks and talking to the neighbors in their life. So your baby is going to be your baby for their life. They're going to need help with everything: eating, using the potty, wearing clothes, knowing what to do, and so on and so forth. So here you can see, in this case, again, lack of some diminishment of pigmentation. And in this case, so this is Colin Farrell, the actor, and he does have a child that has Angelman syndrome. And while he does not release photos of his child or take her out in public, he does help with funding and fundraising for research and other kinds of things related to this. So Angelman syndrome. Is there any chance that he and his wife are likely to, in any further, or partner, I don't know if they're still married, partner, ever have this happen again to any of their children? This is a random mutation, and the chances it would occur again in the family are virtually zero. Just randomly happened, just one of those things that sometimes shows up. In Angelman, again, neurogenetic problems, severe nerve issues from them. And one last term that we learned from today, strabismus. Strabismus is when your eyes, even though you can see, when they don't have the nerve or muscle control to cause them to track parallel ahead in the same plane all the time. So strabismus is the term for if your eyes are crossed, or like this child that you can see at the top, where both are kind of leaning out a little bit, or when one is diverting. Doesn't mean that you can't see. As long as the rest of the message is going through. If your eyes aren't looking in the way that your brain expects, what the brain does is actually decide which one it's going to read, and it just stops being confused after a while. That happens sometimes with some of those eye surgeries. So for instance, if you have real difficulty seeing things that are small, they fix just one eye. So for a little bit after the surgery, it's really confusing if you're trying to look at a distance or look up close, because your brain doesn't know what to do. And then a few weeks in, when you're looking at little stuff, your brain's just really reading one eye. When you're looking far away, it's really just reading the other eye. It never is using the two together because they can't focus on the same stuff. So really interesting stuff. Okay, we got a little bit of time left, and so we will do the first little step here for chromosome numeral abnormality, and then call it a day, because you have plenty to do before I see you next Monday. So chromosome numeral abnormality means that the individual is born with an extra or a missing chromosome. If you have one that's missing, then the term is monosomy, right? Because you don't have it in a pair, you just have one. And if you have an extra, it's trisomy. These are not particularly common for most chromosomes, except for there is one chromosome pair where trisomy occurs fairly more frequently, and the person has a normal lifespan and generally not too many outcomes that are really a big problematic thing for them to divert around as far as they go through their lives. We're going to not work on partial and mosaic right at this particular minute, but I give you 3 ones that we'll look at for a little bit on Monday when we come back. Then on Monday we'll look at the rest of chapter 5. So we're going to look at trisomy 13, which is called Patau syndrome, trisomy 18, Edward, and trisomy 21, Down. So you all, and so many of us, are going to, just as members of communities or perhaps professionally, work with people that may have family members, or at Southwestern College, when we actually get to go to campus, you have a number of coworkers that do facilities work on campus, usually not so much at the time when it's the busiest with the traffic and the students, but around about noon or so, and then through the late afternoon. So I see them all the time, where there are some different types of learning disabilities, but so it isn't always the biggest thing. But when you're reading again about meiosis and the meiosis video, non-disjunction, where does this type of error come from? It comes from errors that occur during gamete formation. So it's not in the parents in most cases. It's just something that happens when the chromosome makes its copy, right? So then it's in the X form with chromatids. And then when it's supposed to let go of the copy, right? The centromere should dissolve and let the copy go. It doesn't dissolve in time. And so one of the cells brings 2 into it, and the other cell is missing that chromosome. So that's called non-disjunction. Okay, this has been pretty intense amount of stuff. I know you have lots to do. Most students have not posted your visual class activity yet today, so please work on that and turn that in by the end of the day. That's worth 10 points. And because it's been up for a number of days, there's no reason to be turning it in late. So, and then be sure, and I'll send you a little nagging, reminding email before the end of the week to come and pick up to come and finish with taking the quiz for that one.